مشاهدي زوم افريقيا، احنا مكملين مع حضراتكم جولتنا في ينجنبي. الحقيقه يعني ما كانش ينفع نكون موجودين في ينجنبي اللي هي كانت اشهر مدينه للابحاث والدراسات الزراعيه والمنتجات والاخشاب من بدايه العشرينات للثلاثينات للسبعينات لحد ما كان في برامج هنا تبنتها منظمه اليونسكو. احنا موجودين دلوقتي مع حضراتكم، هناخدكم في جوله لمعمل خاص جدا للنباتات وبيدرس أعمار الشجر وتأثيرها على تغير المناخ. هناخد حضراتكم دلوقتي في جولة هي معقدة شوية بس هتبين قد إيه المعمل ده مهم لشرح تأثيرات تغير المناخ على العالم كله. خليكم معنا. مشاهدينا، احنا دلوقتي موجودين جوه المعمل لدراسة الأشجار والنباتات النادرة والمهمة جدا ليهم علشان تأثيراتها على إعادة هندلة تغير المناخ. معنا دلوقتي بروفيسور هيبو. Welcome to our program Zoom Africa.
Thank you very much.
So we will start from here, right?
Yes.
So can you explain what that?
Yeah. So what we do is we take samples of wood from the forest to study here in the lab.
Okay.
The first step that we do is next door is in the woodworking workshop. We take small samples, we take them in here. This lab is the wood preparation lab, the sample preparation lab.
Okay.
So we take small blocks of wood, and then we put them on the microtopes.
Here.
Yes, this one or this one, and there's another one there. Different kinds of microtopes.
Yeah.
To make very very thin sections of the wood, microscopic thin sections of the wood, so that we can study those under the microscope.
Okay.
So after we cut the small thin sections, we first bring them to the different baths to dehydrate them, to give them a color. We can give them blue color, we can give them pink color. These different colors are to differentiate the different cell types.
And different result.
And different cell types, so that we can easily see this is this cell type, this is this cell type. The result of which you can see here. So what you see here are microscopic thin sections. So all the wood samples are fixed on small glass plates that we can put under a microscope. So when after this is done, we go to the microscope.
Yeah.
At the microscope, we are going to study how a tree grows, how fast it grows, but also in exactly which time of the year the tree grows. Okay. But then also when exactly the moment is that the cells start absorbing a lot of carbon. Yeah. So when the actual wood is being formed, because there is a difference between when a tree grows and forms very light cells with almost no content, and in a later stage those cells are being filled with wood. Okay. So we are trying to sort out when exactly that happens, and this is very important information for the flux tower that we have here in the forest, where we are measuring when carbon is being taken up. We compare that with permanent inventory plots where we are monitoring the tree growth, but we are going to see that there is a difference between what the flux tower measures when the carbon is being taken up as according to the flux tower and the tree growth. Okay. And that difference is probably because of this difference in growth. The trees are growing, they make cells that contain almost nothing, and it's later on that the cells take up carbon, which is what the flux tower records. Okay. And to really sort out that if that is really because of this differentiation in processes in a tree growth, we have to do this work. Okay. So this is connected to the work that we do in the forest.
Okay.
Yeah.
There is another room we will see.
There is another room over there. That's where we study the thin sections under the microscope.
اوكي مشاهدينا، هنروح للغرفه الثانيه هنشرح اكتر هم بيستخدموا ازاي الاشجار ان هم بيقيسوا اعمارها وتاثيراتها في ان هي بتسحب ثاني اكسيد الكربون وده تاثيره على تغير المناخ. خليكم معانا، هنخش الغرفه الثانيه دلوقتي. مكملين مع حضراتكم. احنا دخلنا دلوقتي الغرفة الثانية و بروفيسور this is type of food.
Yes.
So you can now explain.
Yeah.
Okay.
So this is another kind of research that we are doing, and so we are sanding the surface of this kind of stem discs, big ones, small ones, everything that we find.
Yeah.
And by sanding, we visualize. It becomes clear what the tree ring, the growth ring pattern is. Yeah. And under the microscope, what we're doing here is we indicate the boundaries of each tree ring. And with this device here that is coupled to the microscope and to the computer, so we can actually directly see what we see under the microscope. We can also directly see it on the computer. Yeah? So it's all connected to a computer. And we can actually measure the growth rings by using this device. So here with this device, the computer tracks how fast and how much we move.
Okay.
And we can indicate on the computer each time when we see a growth ring, and the growth rings are being measured automatically with this down to the micrometer.
Okay.
Yeah. And we use these patterns of tree rings, ring widths, to compare with the environmental variables. For example, temperature, precipitation, but also CO2 concentration, to see if there is an influence of these environmental variables on the growth of trees.
Okay,
yeah.
That's fine. Okay.
So we, by just counting the number of rings, we can also estimate how old a certain tree was. And one of the discoveries that we did recently is that the big trees in the forest, they seem to be very old, but they are typically the trees that grow fast. So we found big trees that are about 200 years old. But what we discovered is that if you look at the very little ones that are underneath the canopy, that live in the shade, they have another life strategy. They don't grow fast. They are in a dormant state almost all of the year. Yeah. If you put them under the microscope, you see a lot of very small tree rings. They almost don't grow, and those small trees underneath the big ones can be much older than the big ones.
Ah, okay.
And so that's
strange.
It's not strange. It's just different kinds of life strategies.
Yeah.
But it's important for us scientists and also for forest managers to know that the big trees, they are important for big carbon stocks. Yeah. But the small trees underneath the canopy, they are important for continuity of carbon stocks. Okay. They are important for the long-term carbon capital in a forest. So that is what we discovered by counting tree rings.
Okay, that's fine.
Every ring, it's like a year.
If you look closely, you can see. Yes. So we do a lot of research on which species form annual rings. Not every tree species forms annual rings. Yeah. But this one is called Pericopsis elata. It's known as Aframosia. It's a very well-known timber. Yeah. It's also protected by CITES. And we do a lot of research on it, first of all because it is an emblematic timber species. We need to understand its ecology, its growth, and how its growth is related to climate, to see whether the climate in the future will still sustain its existence. But next to that, it's an interesting species to us because it does form these annual rings. And we have been able to show that because we sometimes take small samples to send to a lab for radiocarbon dating, so that we can actually measure the age of the first or the second ring, and compare that to the age that we estimate by counting the tree rings.
It's very complicated. Yeah.
It's all complicated research, yes. Yeah. But so we organized, we started doing all that in Belgium. We took a lot of samples to Belgium, and our Congolese PhD students needed to come to Belgium to be able to study the wood. Yeah. But now we decided to organize, to install a laboratory here in the middle of the forest, so that we can study ecological characteristics. We can study forest ecology, wood ecology within the rainforest. Oh, that's and more Congolese students can take advantage of the setup. So at the moment we have about 10 students who are working here.
Congolese?
Mostly Congolese students. Yeah. Sometimes you also have master thesis students who come. Right. Sometimes you have Belgian PhD students who come and do some research here. Yeah. But most of them are Congolese PhD students. And the responsible of the lab, the one who is responsible of the lab is one of my PhD students. Oh, okay, so it's actually being run by a PhD student. Yeah. And they do very well.
Oh, that's really amazing.
So it's partly independent. Yeah. But of course they depend on us for consumables. For example, if some of the staining agents are out of stock, then we send more equipment and more consumables. So that they can go on.
Very fine.
And they sometimes come to Belgium for training. Yeah. And of course, they also come to Belgium for to get close to the professor, so that we can work on publications and we can work on their PhD thesis. And they come to Belgium, for example, to defend their theses. So yeah, there's a lot of collaboration, and sometimes we come down here to help them out. We also sometimes organize trainings here within the lab. At the moment, for example, we are training 3 people from the local INRA research institute to become assistants here in the lab. So we are working together with INRA as well.
Great.
We are integrating INRA agents within the functioning of the lab.
مشاهدينا، زي ما شفنا مع حضراتكم احنا موجودين دلوقتي في معمل الأخشاب اللي موجود جوه قلب غابة ينجنبي. يعني شفنا قد ايه المعمل ده معقد ومهم جدا للحفاظ على البيئة. خليكم معانا فاصل، رجعنا لكم تاني.